Hey everybody, so we are moving into chapter 1, the awesome chapter 21, but also probably the one of the most complex chapters we go into. Not necessarily because its material is hard, but essentially I'm trying to explain a process to you when all these pieces are moving together as one. I gotta find a way to teach them to you in bits and pieces as a stepwise process. So essentially we're gonna learn these different processes, and then I'm at the end of this lecture, I'm gonna be like, so all this happens all at once. Okay? So chapter 21 is the immune system. I'd say for exam 2, this is the one that gets students the most, just because again, it's such a complex process. So our plan for today is we're going to go over the innate defenses. We're going to talk about what the innate defenses are. We'll talk about surface barriers, internal innate defenses, cells and chemicals. And then we move into our adaptive defenses, and we'll talk about antigens, where hopefully that word is familiar to you guys. But we'll talk more extensively about antigens. We'll talk about cells of the adaptive immune response, our humoral and our cellular immunity, our immune response, and then we'll talk at the end of this chapter about some homeostatic imbalances of the immune system. So in this first video, we're going to focus on innate defenses. Okay, here's our list of objectives. Again, make sure you guys look over those objectives as you guys study the material for the quizzes and the exam. So we talk about the immune system. It is a functional system rather than an organ system, and what we mean by this is it's basically going to occur systemically throughout the body. It doesn't necessarily follow the path of specific organs. It's going to be throughout the entire body. However, it is comprised of two kind of umbrella systems. One is our innate defense system, and this is nonspecific. And what we mean by nonspecific is, again, the immune system is there to attack and activate a whole cascade of events in response to things invading our bodies. Okay? Now, the innate is nonspecific in that it doesn't care what is entering the body. It doesn't care what kind of arms or what kind of legs it has. It just knows that it's foreign and it's going to elicit a response. Okay? The adaptive is specific. Okay, now I know you guys, I know pathogens don't have arms and legs, but go with me for the sake of this chapter, okay? So the adaptive, on the other hand, not only cares about what the arm looks like, but it also cares about how many fingers it has and what nail color is on those fingers, okay? So it is a very specific response. Okay, and again, when we talk about specificity, we're talking about the foreign substance entering the body. Our adaptive is only going to respond very specifically to those foreign substances. Okay, so the innate responses release proteins that alert cells of the adaptive to these foreign molecules. So you can kind of think about it as the innate is going to warn the adaptive of the impeding danger. Okay, now, as we move through the lecture today, foreign substance, oh my gosh, if I can spell, is going to be the same as a pathogen. And pathogens can be bacteria, virus, parasites, things such as dander, anything foreign. Okay, so when pathogen is typically a disease-causing agent, however, in the sake of the immune system, I will often use pathogen as just a foreign substance. Okay, if you're taking microbiology right now, you know that that's not a true statement, but you get the gist of what I'm trying to say. So when we look at our different kind of defenses, we have, again, our innate defense. We have two lines of defense within our innate system. We have our surface barriers. This includes things such as our skin and mucosa, which we'll talk about more extensively here on the next slide. Our second line of defense are our internal defenses. So if our skin or our mucosal barriers become breached, we have these internal defenses that, again, remember, this is a non-specific response. It doesn't care what's come in. It just is like it's sounding off the alarm to the body, okay? And our internal defenses include things such as phagocytes, natural killer cells, inflammation, antimicrobial proteins, and fever. And we're going to talk about each one of these individually. And then our third line of defense, okay, and this should be more kind of over here, because not necessarily our humoral immunity isn't necessarily our third. Our adaptive, if I can get my eraser to work, our adaptive responses are really kind of our third line of defense. And this involves two arms, our humoral immunity, which is our B cells, and our cellular immunity, which are our T cells. And don't forget, when we talk about B cells, these are going to be B lymphocytes. Okay. When we talk about T cells, these are our T lymphocytes. Okay. And both of these systems activate simultaneously, but they only attack particular foreign substances. Okay. So again, this is a very specific response, whereas our innate is a non-specific response. So what we're going to do is we're going to first focus on our innate defenses. They're kind of the easiest to explain, they're the easiest to kind of understand, and then we get into the more complex adaptive defenses. So our first line of attack are our skin, right? Our skin serves as a perfect protective mechanism. And one of the reasons why our skin serves as a protective mechanism is because our skin actually has a slight acidity to it. Okay. And the secretions that are the substances that are secreted onto the surface of our skin, because they have that slight acidity to it, this inhibits the growth of pathogens on our skin. Okay. So when the pH of your body gets slightly off, that allows certain pathogens to proliferate. So for example, if you think about yeast infections, okay, so some women are susceptible to yeast infections. Athlete's foot is a fungal infection. Typically what happens is because those become dry or become super sweaty regions, it can slightly change the secretions in that region and cause the acidity to change, and then you allow for microbes, pathogens to kind of proliferate. In addition, so when I say skin, these are mucosal membranes and their secretions. In addition to that, we also secrete enzymes. So for example, in our saliva, which is going to be produced by our mucosal membranes, we secrete lysozymes. Okay, these lysozymes will basically destroy pathogens as they try to get through our respiratory or through our oral cavity. Our respiratory mucus, right? Those mucin cells that are secreted within the epithelium of our respiratory system are going to contain lysosomes that will also attack and destroy pathogens as they try to enter, as well as our lacrimal fluid. Remember, that's the fluid that comes in our tears, right? We talked about that in AMP1. And again, all of these will kill these microorganisms, okay? The third way in which our skin or these surface barriers serve as a protective mechanism is through the secretion of peptides. And all a peptide is typically 2 amino acids linked together. So a protein could be hundreds of amino acids. Peptides are usually much, much smaller little teeny fragments of proteins. But anyways, we have these antimicrobial peptides called defensins, and that's actually what they're called. And these will inhibit growth within our surface barriers. The last one is mucin. Again, I mentioned this because mucin kind of plays a role when we talk about this respiratory mucus. But mucin is produced by those goblet cells or those unicellular glands. And when it combines with water, it forms mucus. But what this does is it traps. It basically becomes like a fly paper, right? A sticky trap that traps these microorganisms that then prevents them from being able to invade or get past that first line of defense. However, every once in a while, that first line of defense becomes breached, whether through a cut or through the respiratory system, through the oral cavity, the nasal cavity, some way or another, that pathogen is able to breach that first line of defense. So what happens is once that pathogen has breached that first line of defense, we need to have mechanisms available to kind of fend off as best we can the pathogen that then enters our body. So we have various ways or various mechanisms in our second line of defense that will help us kind of combat the entry of these pathogens into our body. So we have phagocytes, we have natural killer cells, we have inflammatory responses, and we'll talk about the different types of inflammatory responses, and we have antimicrobial proteins. More specifically, we're going to talk about interferons and complement. And complement is a group of proteins that become activated, and again, we'll talk about that here in a little while. So let's first talk about phagocytes. So we know from our blood chapter, chapter 17, that neutrophils are super phagocytic, okay? They are the most abundant of all of our phagocytes. However, although they're super important and they're awesome at what they do, they die in the fight. Okay? So this is probably why we have so many neutrophils, because they can't survive and kill. They can't kill and survive at the same time. So what happens is a neutrophil's cruising around, okay? When it becomes exposed to a pathogen or into an infectious agent, it actually transforms itself into a phagocytic cell and engulfs that pathogen or infectious material. And I feel like I don't really know what phagocytosis is. We're going to get into it on the next few slides when we talk about our macrophages. So macrophages, okay, are actually developed from our monocytes. So here is our awesome monocyte. And when that monocyte leaves circulation and enters into the interstitial kind of space around within our connective tissue or around our cells of our epithelium, they will transform into a cell known as a macrophage, okay? And often I will write the abbreviation. Often macrophages are kind of written like that as their abbreviation, okay? This is the chief phagocytic cell. And how they work is quite spectacular. So here is a macrophage. So this cell right here, this kind of purple cell is a macrophage. And these green little rods, these are bacteria. Okay. And so what happens is this macrophage sends out these cytoplasmic extensions and essentially binds to the bacteria in the environment. Okay. And then what it does is it basically retracts back those cytoplasmic extensions, bringing the pathogen to the surface of the cell. Now, unlike neutrophils, macrophages do not die in the fight. And we're going to see why that is here in a minute. Right, so here's, for example, a cytoplasmic extension. We bring it back. Here's this bacteria right here. And we're going to envision that this is the bacteria over here. Okay. And what happens is this macrophage will actually, through endocytosis, okay, through an endocytosis process, engulf this pathogen, okay, this bacteria or whatever it might be. It forms these little things called pseudopods, okay? And what happens is once this pseudopod kind of cuts off, it forms something called a phagosome. And a phagosome is a phagocytic vesicle. Okay, so now you have this pathogen or this infectious material inside of a vesicle. Okay, now inside of this macrophage, it also produces vesicles that are coming off the Golgi apparatus known as lysosomes. Okay, and lysosomes are going to contain a whole bunch of hydrolytic enzymes, okay, or enzymes that are basically going to destroy things. So what happens is this lysosome merges with this phagosome and becomes a phagolysosome. Yeah, I know. Crazy. Okay. And what happens is when this merges, the enzymes that were located in this lysosome combine with the phagosome and ends up destroying the foreign substance or the pathogen or the whatever the foreign substance is. Okay. And when it destroys it, it doesn't just release it to the inside of the cell. What it does is it comes down this phagolysosome that now has all of this digested particle in it, comes down and through an exocytosis process spits. I know spits is not the proper word, but removes, releases undigestible material out to around the cell. Okay. Now, I want you guys to remember this stuff. You might be like, oh yeah, we're just getting rid of it. Oh, this becomes so important. I'm just going to put a little asterisk here so later, when I come back to this and be like, remember that asterisk I drew around that population of spit, right? You guys will remember what I'm talking about, okay? Now, the importance and the reason why this macrophage doesn't die in this process is because of the fact that it stays in basically within this vesicle. The digestive process stays within this vesicle. If, for example, you consumed this macrophage, consumed this foreign substance, but didn't maintain it in a vesicle, okay, and it just released its lysosome, the enzymes within the lysosome into the inside of the cell, that lysosome would, or the enzymes within that lysosome would destroy all of the organelles in the cell. So it would kill the Golgi and the ER, the rough and the smooth ER, the nucleus. It would destroy everything. So by maintaining itself within these vesicles and the formation of this phagolysosome, this monocyte or this macrophage is able to survive this phagocytic process. Pretty freaking cool. Okay. So that was one type of example of our, and so again, this isn't going to be like one or another. So a lot of these things will all happen. Okay. Our second type of second line of defense are natural killer cells. Now, one major difference between a monocyte or a macrophage and a neutrophil a macrophage and a neutrophil's ability to phagocytize and what its function is, and a natural killer cells is the macrophage and the neutrophil attack bacteria that have not entered cells. Okay, because remember, we're talking about a pathogen that has basically breached the surface, whether it be the skin or the mucosal membrane. But because they've breached that doesn't mean it's gotten into a cell. It just means it's cruising around the interstitial fluid or the interstitial space around the cells or within the connective tissue. Just because we've breached the first line of defense doesn't mean that pathogen has gotten into a cell. Okay? However, if the pathogen is able to enter a cell, this will activate the natural killer cell. So the natural killer cell is a non-phagocytic cell. It's a large granular lymphocyte. It's a type of lymphocyte. Okay. And they attack cells that lack cell surface receptors. Okay. So let's think about this. This is probably one of the, I don't know, I think these are just so freaking cool. So here's our natural killer cell. This doesn't come out of your guys' textbook. This is from a different textbook. I was trying to find a good visual so you guys could understand this process. Okay. So here's our natural killer cell. Okay. Here's the cell of our body. Every single cell of our body has this type of receptor on it. Okay. And we'll learn later what this receptor is. So just for now, just know that it's like waving at the natural killer cell, like, hey, it's me. I'm supposed to be here. I'm healthy. I'm good. Just keep moving on. Okay. That's this guy, right? This is the I'm fine and healthy flag. Okay. So the natural killer cell says, okay, good, cool. High five. I'm moving on. Okay. Now, a virus enters our body. Now, viruses are notorious for entering into the cell itself. Typically, bacteria don't enter into the cell, but viruses do. And that's because they have to hijack our machinery in order to carry out their survival. So, a virus enters the cell, okay? And this cell that was once healthy says, oh my God, I've been infected. And what it does is it removes those flags. It says, I'm going to let the virus think that I'm a healthy cell, and I'm going to remove the arms that are like, hi, I'm fine. And what happens is when the natural killer cell comes around, it's cruising around, all of a sudden it comes to a cell and it says, oh my God, where's its hands? They've disappeared. And once they've disappeared, this natural killer cell knows to destroy this host cell. Isn't that so cool? Right? So again, natural killer cells will only do this if a cell in the body has become infected. Okay? So what it does is in this process here, where it sees an infected cell, it releases chemicals that initiate apoptosis. And apoptosis, if you're not sure what that means, this is often referred to as self-suicide. So essentially it turns on genes, okay, within our DNA that elicit kind of like a bomb response within that cell. So that cell actually triggers cell death. Okay. And again, this will happen if it's infected with a virus, that cell, or that cell becomes cancerous. A lot of the time that self marker disappears. Super cool. The third way which internal defenses activate, okay, is through our inflammatory response. So now again, we breach the first line, okay? The phagocytic cells are going to start fighting unless we're a virus, and that virus gets into a cell, and then our natural killer cells will start fighting. At the same time, we're going to elicit this inflammatory response. Okay? So again, it's triggered anytime the body's tissues are injured. And what this really does is prevents the spread. It attempts to prevent the spread of any damaging agents that have just breached that first line of defense. So the inflammatory response will dispose of cell debris as well as pathogens. It'll alert the adaptive immune response, right? That other arm, the one that's way over there that we haven't talked about yet. Okay? But it's also going to set the stage for repair. So as much as inflammation sucks, okay, it's actually good during certain times. Like, we want an inflammatory response if we have some type of invasion, okay? And so when we think about inflammation, again, a lot of these things, right, you get a cut in your arm, right, you get the redness, it gets warm to the touch, you get a little bit of swelling, and it's painful, okay? And the reason for that is because of this really big, complex process that is occurring, okay? So what happens is, you get this initial stimuli. You get this tissue injury, right? You get this bloody finger, you cut yourself on a piece of glass, okay? And the first thing that's going to happen is you're going to release, or your body is going to release massive amounts of chemicals, okay? Now, we have these two arms, and I'm going to hold off on this arm over here. I'm going to kind of go through this arm first. But remember, these are both happening simultaneously, okay? So we release these inflammatory chemicals, things such as histamine and complement, which we'll talk about here in a while, kinins, prostaglandins. Again, you don't need to know the specifics of these, but we release these are all inflammatory chemicals. And what they do is kind of a very broad range of effects. So, for example, this release of inflammatory chemicals will cause our arterioles to dilate. Okay, that means we're going to increase blood flow. Okay, this will cause this local hyperemia, which is an increased blood flow to the area. Okay, this causes the heat. Okay, And that's because we have an increase in metabolic activity of the cells, the new cells that are coming in. They're starting to eat up the cellular debris, and they're trying to find the pathogen and eat the pathogen. Okay. In addition to this is where we get the redness. Okay. So that local hyperemia is the source of the heat, is the source of the redness. Okay. Additionally, these inflammatory chemicals will directly cause activation of our pain receptors. Okay, so they actually they bind to the neuron receptors and trigger that pain response. We get increased capillary permeability. Okay, so remember, our arterioles are controlling the flow in. So not only do we increase the flow in, but we increase the seepage out of that capillary. That seepage out, okay, which is called exudate formation, that's where we're getting that swelling. And in addition to that swelling, right, triggering pain, okay, because the swelling, it causes pain, okay, it's also going to basically, potentially, depending on what's damaged and how bad it's damaged, will cause a temporary impairment of function of that region. The last thing that's going to do is it's going to attract neutrophils, monocytes, and lymphocytes to the area. And this is through something called chemotaxis. Okay. Now, what you're going to see is that this kind of area over here crosses over to over here. And this process over here is called phagocytic or phagocyte mobilization. Okay, and encompasses kind of this and this visually. So kind of the words are here, and you can kind of visually see it here, right? So we have our tissue injury, okay? And what happens is, again, chemicals are released, okay? These chemicals are released from the injured tissue, and this promotes leukocytosis, which is the release of neutrophils from our bone marrow in response to these chemicals being released. Okay. And what happens is once we get leukocytes, again, that's going to include our monocytes, our neutrophils. Okay. Once that happens, these white blood cells will start cruising along the bloodstream, and they're just like, where is it? Where is it? Where is it? Right? And as it gets closer to the site of injury, the chemicals that are being released by the tissue that's damaged will start to cause these leukocytes to kind of attach themselves to the edge of the capillary wall. This is something called margination. Margination is essentially where the neutrophils and the monocytes actually start to cling to the wall in preparation of being able to exit out of the capillary system in a process known as diapedesis, okay, where those leukocytes are able to basically pass through the capillary wall and into the area of damage. Okay. Once they're in that area of damage, they get to chow down. That neutrophil is going to start phagocytizing that region. Okay, the neutrophil will come in first. That will then be followed by the monocyte, i.e. the macrophage. So what will happen is the neutrophils will try to elicit or try to basically control the army of pathogens being able to penetrate this area. And then when the macrophages come in, they come in and clean up the neutrophils and also start at the attack as well. Okay, so again, a really cool process, but a really important kind of process of how the inflammatory response is a major player in our kind of controlling that pathogen dispersion, or preventing that pathogen from moving around the rest of the body. The last kind of mechanism we have in our second line of defense are our antimicrobial proteins. And we have two types of antimicrobial proteins I'm going to talk about. One are interferons, the other is complement. So when we talk about interferons, these are an umbrella term used for a large family of proteins that exist within our cells. And interferons, often referred to as IFNs, How they work is they typically are going to work in response to viruses. So like the natural killer cell, these interferons are going to work in conjunction with the natural killer cells. And what happens is, under a normal circumstance, a virus will inject its genetic material into a cell. And it will utilize, okay, the machinery of the cell to make more virus. Okay? However, when that virus enters the cell, in addition to hijacking the machinery, it also activates the interferon genes of that cell. Okay, so this is kind of a consequence of the virus entering that it hasn't figured out how to, well, most viruses haven't figured out how to turn off. Okay, so what happens is this virus comes in, gets to produce more virus. It's like, woohoo, I'm producing more virus. Unbeknownst to it, this first cell is also making proteins, okay, interferons, to warn its neighbors that it has been infected. Okay, so the cell produces these interferons, releases them via exocytosis. Okay, and what happens is now is this interferon signals to its neighbors, okay, to make an antiviral messenger RNA, which ends up making proteins that block viral reproduction or viral copying. Okay. Now again, this relies on this first cell being able to be activated, or the interferons being activated, in order for it to warn those neighboring cells. Okay. Now, there's a number of artificial interferons that have been developed by pharmaceuticals over the years to treat a number of different types of viruses. So, for example, hepatitis C, genital warts, multiple sclerosis, hairy cell leukemia, which is a type of leukemia. So you can make these in the lab and give them via an injection into a person, or intravenously into a person, and have those protect against viral replication. Another antimicrobial protein system is called complement. Okay, now complement is the system. It's a horrible name. Okay, the complement. So just like, you know, we talked about blood clotting, and blood clotting being more specifically hemostasis, that process of clotting. Complement, okay, is a group of proteins. Okay, so it's about 20 blood proteins that circulate in an inactive form. Unlike hemostasis, we're not going to go into the cascade of how they become activated. Okay, but these complement proteins, when they become activated, okay, are a major mechanism for destroying foreign substances. Okay, and so what I do want you to know, okay, is kind of the three consequences of complement activation. Okay, so we get complement activation. So up here in this area, right, we are activating these 20 proteins in a cascade of events. Okay, so once we activate complement, three things are going to happen. One is called opsonization. Okay, so we have this pathogen. Okay, let's see. Here's a pathogen. Oh, here's our pathogen. And I like to draw pathogens with little things like that inside them. Okay, so in this case, this pathogen is going to be coded by this complement molecule, C3b. And what it does, let's see if I can get my black, it's going to coat that pathogen. Okay, this is in a process called opsonization. So the first thing that's going to happen is opsonization. Okay, again, the coating of this pathogen. When opsonization occurs, when this complement molecule encompasses this pathogen, It is like a flashing signal to our phagocytic cells, our macrophages, our neutrophils, like, hey, come get this guy, he's bad. Okay? The second thing that's going to happen as a response of activating complement, okay, is complement through C3b or C3a, okay, will stimulate histamine release, which increases blood vessel permeability, which attracts phagocytes by chemocytosis, etc., etc. So we go back through that whole inflammatory response, that phagocytic mobilization process we just went through. That's what this is going to activate. Okay? So our second thing is we're going to increase inflammation. Okay? And our third thing is we are going to directly destroy the pathogen. And how we do that is this molecule right here, and again, you don't need to know the complement molecules that make this up, essentially forms or creates a funnel-like structure. And this is called the MAC complex, or I call it the MAC molecule. Okay. And this molecule then inserts itself into the membrane. Okay. And what happens is this creates pores throughout the membrane and causes a massive rush of fluid inside into the cell and causes it to lyse. So this causes lyse. And if you want to be really non-scientific, lysine is essentially the rupture. Rupture. Oh my gosh. Rupture of that cell. Okay. So those are the mechanisms in which our innate defenses carry out their function.